to explain ways that organisms use released energy for maintaining homeostasis. This is a process called active transport, to use energy to maintain homeostasis. And then the essential question that we seek to answer by the end of today's lesson: how does the cell membrane facilitate the transport of materials into and out of the cell? What I've noticed in my conferences with students, not only from this class, but from my standard class as well, the students who are taking really strong notes tend to be the students who are doing well because they're having an easier time completing the asynchronous assignment. They have opportunities to review better for the tests. And so all of these things really rest on the foundation of taking strong notes, paying attention during class and writing down strong notes. Even if you have to type them up, take some type of record of what we're talking about. Janelle, I'm not sure what you mean by that.
Like the stuff that you doing now. She already taught.
What class was this?
It's the same class.
The same? You were taking another biology class?
No, it wasn't at that school.
Oh, okay. And Lance, I want to say I enjoyed that video of the I can't remember what they actually called them, but the organisms that were living at the bottom of these seas, living off of light produced from some type of process of releasing heat. That was interesting. So photosynthesis doesn't always have to occur with sunlight, but it does occur with some source of light. So that was cool. Thanks for sharing that. Okay. So we did see this table yesterday, but I think we had to move through it pretty quickly. So I wanted to show it again. For those of you who perhaps didn't get a chance to finish writing it down, you now have that chance. And I'm going to talk a little bit more about it as well. So again, the question is, which molecules can cross the membrane easily? The column on the left describes molecules that are able to cross the membrane without any additional help. They just move through the membrane by themselves. These tend to be small, nonpolar, and neutral particles, such as oxygen, nitrogen, carbon dioxide, and CH4, which is methane. So small molecules can fit through those hydrophilic heads and make their way through the phospholipid bilayer pretty easily. They have to be nonpolar though. And the reason for that is a chemical reason, so I won't say too much about it. But the reason they have to be nonpolar is because the hydrophobic region of the cell membrane is also nonpolar. So those fatty acid tails that kind of face one another in the inside of the membrane, they're nonpolar. These molecules also have to be uncharged. They have to be neutral. They can't have a positive or a negative electrical charge. On the other hand, other molecules do need some help. They might need to move through a protein channel. They might need the additional help of some cellular energy, which we'll talk about today. But these molecules tend to be larger. They might be polar, or they might have an electrical charge. So some examples include NaCl and glucose, C6H12O6. Water is another example of one of these molecules that needs some help. So this is a great chart to have written down. You can refer back to it for the rest of our lesson today. not going to do that right now. So we talked about it yesterday, but obviously homeostasis is the goal. Cells, they are charged with maintaining an internal balance for the organism. It's the job of each individual cell to contribute to homeostasis. Each individual cell needs to be at some type of balance. They need to exchange materials with their surrounding environment in order to maintain that balance. So they're constantly letting in certain molecules and they're also releasing certain molecules to their environment. That's the simplified version of what's going on. It is indeed a bit more complicated. There are some situations where the cell does not want to be at equilibrium. It will intentionally create a concentration gradient where there might be more of something outside of the cell than there is inside. But in general, we have to think about equilibrium as being the goal in order to maintain homeostasis. So we're going to exchange molecules until they are basically equal with one another, oftentimes. All right. So the first type of transport is called passive transport. Passive transport moves molecules from areas of high concentration to areas of low concentration. So if there are a lot of oxygen molecules outside of the cell and very few oxygen molecules inside of the cell, we have a concentration gradient here. So some of those oxygen molecules are going to move through the membrane into the cell so that we can balance out the levels. We want equilibrium. We want the concentrations to be equal. And that's a passive transport process. Because we're moving from high to low, no energy is required. These processes can take place without the additional input of energy. Just like rolling down a hill. You could literally go to the top of a hill, and if it's steep enough, you could just lay down and you'll start to roll down that hill. No additional energy needed. I want to encourage you all to write down at the very least the words that you are seeing highlighted. All right. So within passive transport, there are a few different types. There's simple diffusion, often referred to just as diffusion. This is the movement of molecules through the membrane without the help of a channel protein. So they literally just move by themselves. They just move through the membrane. These tend to be your smallest, most nonpolar molecules. Oxygen, nitrogen, carbon dioxide, very, very small molecules. They don't need the additional help of a channel protein. Facilitated diffusion, on the other hand, does require the use of a channel protein. And a channel protein is basically just a large protein that spans the membrane, and it's a tunnel, essentially, from the inside of the cell to the outside. And then lastly, there's also osmosis. Osmosis is a very important term. It only applies to the movement of water. Osmosis is a form of facilitated diffusion because water is not able to move through the membrane by itself. Water relies on what's called an aquaporin. It's a very specific type of channel protein that allows for water molecules to move through the membrane. Anytime we see or hear about the movement of water, we should automatically be thinking osmosis. This is probably a term you all have heard before, but maybe just not in this sense. So again, I want to encourage you to write down at the very least the words that you see highlighted here. Okay. So here's a cool animation from the Amoeba Sisters that demonstrates these processes. So simple diffusion is on the left. Those fuchsia colored molecules are able to move through the membrane by themselves. We can see this membrane is a bilayer. We've got the hydrophilic heads that do like water. So they face outwards towards the aqueous environments. We can also see that they've got these hydrophobic region here, the fatty acid tails, the lipid tails. They don't like water, so they face one another internally. They're insulated. They don't ever have to come in contact with water. Also, it's cool that from this animation, we can see they've labeled low concentration and high concentration. You can see that in both cases, the molecules are moving from areas of high concentration to areas of low concentration. That's what makes this passive transport, high to low. On the other hand, high to low. Facilitated diffusion, we can see this looks like a hot dog bun, but it's actually what's called the channel protein. And it's just a tunnel through the membrane that allows the molecules to fit through. So these might be larger molecules, or maybe they're polar, or they have some type of electrical charge. So they need the help of a channel protein to get through the membrane. Okay. I always like to think of this analogy of being on a hill. Hopefully, you all, as children, had an opportunity to be on hills at the top or at the bottom of a hill. You would run up or you'd run down. So, at the top of the hill, that's like the high concentration area. The bottom is the low concentration area. In passive transport, no energy is needed. So, it's similar to just rolling a bowling ball down a hill. If you placed the bowling ball at the top of the hill and just gave it a really small nudge just to get it rolling, gravity is going to do the rest of the work. So a small nudge is going to get this bowling ball all the way down the hill. Pretty easy. The other type of transport, the inverse of passive, is active transport. This is when molecules get pumped against the gradient. And I use that word pumped specifically and intentionally because when we see it, we should be thinking about energy. It takes energy to pump something. And active transport requires cellular energy called ATP. We've talked about this before. So ATP is required to move molecules against their concentration gradient. This means to move them from an area of low concentration to an area of high concentration. It requires an input of ATP. Excuse me.
Can you say that last part?
Say that one more time, Lisa.
Can you say that last part? You were breaking up.
Say the last part again.
Yeah, because you were breaking up on my part.
Okay. I don't remember what the last thing I said was, but I'll just say the whole thing again. Molecules in active transport get pumped against their concentration gradient. So that means being pumped from an area of low concentration to an area of high concentration. And again, concentration, as we talked about yesterday, is just a measure of how much of something there is in a given space, in a given volume. So low concentration means that there's not much of something relative to another region. It's all relative. We're thinking about how one thing compares to another. So if one area has a lower concentration, what we're saying is that there's less of that molecule or less of that material or less of that substance than there is in another region of higher concentration where there's more of it. So active transport, we're going to pump something. We're going to move it from this low concentration region to a higher concentration region. That takes energy, just like going from the bottom of a hill to the top takes energy. Here are some examples of active transport. I will ask that you write down these examples, and I'm going to talk briefly about each of them, but we won't need to go into depth. That's not something that you'll be tested on, but you should at least be able to recognize that when we see these phrases, we know we're talking about active transport. So just write them down, and then I'll talk about them briefly. Okay. So if you didn't finish writing them down, here they are again, but I'm going to go ahead and talk about them. So a sodium potassium pump, this is really a really important feature of cells that plays a huge role in our nervous systems. This is really how our nervous cells are able to communicate with one another. What happens is that with the use of energy, we can see ATP it's small on your screens, I'm sure, so I apologize about that. But we need energy to start this process. This is why it is a form of active transport. We're going to take sodium from an area of low concentration to an area of high concentration outside of the cell. We're going to pump it out. And at the same time, we're going to bring in potassium. This is something, if you're interested in learning more about the physiology of this process and how it contributes to us literally, this is the electrical current that is running inside of our bodies, then you should take AP biology. We talk about it more there. But excretion of toxins, a lysosome is a specific organelle that we don't talk much about in this class. Again, if you're interested, take AP biology. But a lysosome is basically just a big sack of enzymes. And these are hydrolytic enzymes, meaning that they are enzymes that have the job of breaking things down. They literally just go around and they just chew things up, essentially. And so these lysosomes are used to break down things and turn them into waste products. So let's say a toxin enters a cell, the lysosomes will essentially swarm that toxin and try to break it down as best as possible. And then the waste products that are produced from this breakdown then get released from the cell. Endocytosis and exocytosis. This is when the cell membrane essentially pinches itself off to create a vesicle. And a vesicle is kind of just like a transport vehicle for something that's entering the cell or something that's leaving the cell. So small portions of the plasma membrane can kind of pinch themselves off and create this now free-floating membrane to carry something around the cell. Endocytosis is when something enters the cell. Exocytosis is when the cell is getting rid of something. So we can see that in this case, whatever this material is, it formed a vesicle, and then that vesicle fused with the membrane, and in doing so, it released that material to the outside of the cell. All of these processes require energy. They require ATP, so they are active transport. Active transport is moving from a region of low concentration to a region of high concentration. So it requires energy, just like if you had a bowling ball at the bottom of a hill and you were trying to kick it up the hill, it's going to take a lot of time. You're going to need to use a lot of carbs. Why would you need to use carbohydrates? What do carbohydrates provide?
Energy.
Excellent. Thank you. I don't know who said that. I couldn't catch it, but I appreciate you. Yeah, carbohydrates provide short-term energy. Glucose is what we actually use to make ATP. So it gives your body the opportunity to do cellular respiration. You use that glucose to make more energy, to make ATP. So we're starting to wrap up these concepts, guys. We're starting to make connections. And I really want to think about a potential flow chart that maybe we should do that as a part of our class tomorrow. Try to draw a flow chart that starts to connect all of these different concepts that we talked about over these first 4 units. Or maybe we'll save that for unit 5 just before the midterm. But I like to think about this as the 3 A's. A for active transport, A for ATP because you need ATP to do active transport, and then A for against the gradient because active transport takes things from areas of low concentration to areas of high concentration. So those are the 3 A's. And if you can remember that, it will definitely make it easier for you to start to identify different types of transport. So passive transport is like this. You're just kind of a part of this crowd. It's moving from the really crowded area, the really dense area, to the area where there are no people. You basically get carried by this crowd if you really wanted to. You wouldn't have to do much moving at all. They're going to push you. So it doesn't require much energy. But if you were to go the other way, let's say you were an employee trying to get to the outside, it's going to require a lot of energy to kind of dodge all these people. They're pushing and shoving past you. So you've got to push and shove past them. So active transport is kind of going against the gradient from a less crowded region to a more crowded region. It takes a lot of energy to do that. All right, so what we have here is another kind of illustration that I made. You can see that we've got the inside of a cell. We see the cell's membrane, this brown thing. We see we've got a couple different molecules. We've got O2, which is oxygen gas, We've got CO2, which is carbon dioxide gas. We can see that there's a concentration gradient here because we've got unequal amounts of both. Inside of the cell, we have 6 molecules of oxygen gas, but outside of the cell, we only have 3. Inside of the cell, we've got 2 molecules of carbon dioxide gas, and outside of the cell, we have 7. So there's a concentration gradient here. There's an unequal amount. So my first question, in order to achieve equilibrium, in what direction will the oxygen molecules move? If they're trying to achieve equilibrium, will they move into the cell or will they move out of the cell? Okay, Abigail says out. Melissa says out. Anybody else have a thought? If not, then can I ask one of you two to explain why you say out? How can you tell that that oxygen is going to move out of the cell? Yeah, because there are more molecules of oxygen inside than there are outside. So in order to get to an equal number, they're going to have to move out. We've got 6 inside and only 3 outside. So let's move 1. Now we've got 5 inside and 4 outside. So still not at equilibrium. So let's move this one. Now we've got 4 inside and 5 outside. So what we're seeing here is that we can never quite be equal. What's going to happen is that this oxygen is going to have to keep jumping back and forth. It's going to jump back and forth. This is a phenomenon called dynamic equilibrium. The word dynamic means changing or moving. We refer to this as dynamic equilibrium because yes, we are establishing a balance here, but we can never just be at a stable balance. We can never have 4 and 4 or 5 and 5. In order for us to maintain a balance, one molecule is always going to have to be shifting back and forth. And so we call it dynamic equilibrium. So what about the other molecule? In what direction will the carbon dioxide molecules move? Into the cell or out of the cell? Thank you, Janelle. Yes, they'll have to move in. We started off with 7 on the outside of the cell and only 2 on the inside. So that one's got to jump in. That one's got to jump in. Now we've got a situation where we would have 4 and 5. Again, we're going to have to go through this process of dynamic equilibrium where one is shifting, is moving in and out. So my question now, what kind of transport just took place? Was it active or passive transport? Was that active or passive? Okay, let me hear from one person who said active. Why do we think active? Okay, why did you think it required energy? I didn't see. I didn't mean to imply that there's no ATP here, so I didn't mean to imply that it was requiring energy. Remember the difference, another difference, you're right, Janelle, to think that, one of the differences is that active requires energy, passive does not. But here there's no mention of energy. There's no mention of ATP. So that doesn't really help us to determine what kind of transport it is. The other thing we can think about is it moving from high to low or is it moving from low to high? If it's moving from an area of low concentration to an area of high concentration, then that would be active transport. But in this case, we had both molecules move from areas of high concentration to areas of low concentration. For example, we started off with 6 oxygen molecules on the inside. That's a high concentration, and 3 on the outside. That's lower. And they moved to the outside. So they moved from high to low. They moved from high to low. So that's a passive transport process. Same thing with carbon dioxide. We're starting off in a region of higher concentration. There are 7 carbon dioxide molecules outside of the cell and only 2 on the inside. So we're going to move from an area of high concentration to an area of low concentration, high to low. That's called passive transport. And because these are specifically small molecules, this is actually more specifically simple diffusion. They don't need the help of a protein. They just move by themselves across the membrane. So this is passive transportation. Passive transport, simple diffusion. So if this is still a little confusing, I understand. I think the asynchronous assignment is going to really help you all kind of think through it. Hopefully you took notes and now you can start to, as you do your asynchronous assignments, maybe you add on to your notes or you clarify your notes or you add an image or two. It's always a good idea. The asynchronous assignment today is U4D3 cellular transport practice. We do have those other 3 that have been previously assigned on Friday and Tuesday that I don't want you all to forget about. There is also an exit ticket. We've got about 20 minutes left, so there should be plenty of time to go ahead and knock out today's assignment as well as the exit ticket. Today's assignment is just multiple choice questions, no free response, so it should be pretty quick. And then the exit ticket, per usual, is just 4 questions. So I'll be here on mute. Go ahead and knock one of these or both of them out. Question 9 of today's assignment, Janelle.
Yeah.
All right, give me a second. Okay. So, what are you not understanding about it?
All of the triangles look the same.
Yeah, they look the same because they are the same. So they all represent glucose. Um, what's happening here is that we've got a cell with what appears to be. What'd you say? Oh, I thought okay. So we've got a cell that has 3 glucose molecules inside of it, and we've got outside of the cell in blood, which I guess is the external environment, we've got 9 glucose molecules.
I understand the blood part. I thought it was like 2 different types.
Nope, same type, all glucose.
Okay.
Okay, folks, so it is now 1:20. So you all are good to go ahead and enjoy your next 10 minutes before your fourth block class begins. I appreciate your time today, and I look forward to talking to you all on Wednesday. Tomorrow is a half day. Keep that in mind. So we're meeting earlier